Chapter 4. Cell membrane transport. 1. Transport mechanisms can require energy or not require energy. 2. There are passive transport mechanisms. 3 types are simple diffusion, facilitated diffusion, and osmosis. 3. Passive transport mechanisms do not require cellular energy or ATP. 4. Active transport mechanisms always require cellular energy or ATP. 5. Active transport mechanisms across the cell membrane has 2 types of cargo. 6. When the cell transports solutes across the cell or plasma membrane, it can use single diffusion or facilitated diffusion with passive transport, or primary or secondary active transport or vehicular transport with active transport. 7. Osmosis means the transport of a solvent across the cell or plasma membrane, with osmolarity or tonicity. In the context of cells, solvent means water. 8. The difference between passive transport and active transport compared to osmosis is osmosis transports a solvent across the cell or plasma membrane, whereas passive and active transport only solutes across the cell or plasma membrane. 9. Simple diffusion is one of the 3 passive transport mechanisms, driven by concentration gradient. High concentration moves to low concentration. 10. Transmembrane proteins are not required in simple diffusion. 11. Concentration gradients are examples of chemical driving forces. 12. When equilibrium is reached in the concentration gradient, there is no more net flux. Net flux equals 0 when both sides have the same concentration. 13. Cell or plasma membrane is built from phospholipids. The solute to be able to move through the cell membrane has to be fat-soluble solutes to be moved through the fat-based cell membrane through simple diffusion. 14. Simple diffusion transports only fat-soluble solutes across the cell membrane using the concentration gradient. 15. Channels are examples of transmembrane proteins. 16. Hydrophilic means water-soluble or water-loving. 17. Hydrophilic solutes cannot cross the cell membrane because it is a phospholipid layer, unless it goes through a transmembrane protein. 18. Small hydrophilic solutes are sodium, potassium, calcium, all cations, cannot cross the cell membrane phospholipid bilayer. 19. Transmembrane proteins in the cell membrane are channels that have a passageway allowing small hydrophilic solutes to flow through it into or out of the cell, moved by the concentration gradient. 20. Large hydrophilic molecules like glucose cannot fit in the transmembrane protein pore. 21. Facilitated diffusion is a different passive transport mechanism, still utilizes the concentration gradient, but also utilizes the transmembrane protein channels for small-size hydrophilic solutes to pass through. 22. Transmembrane proteins are required in facilitated diffusion. 23. Transmembrane protein channels are selective to specific ionic species. Some allow cations to flux, and others that allow anions to flux. 24. Facilitated diffusion via carriers, transporting large hydrophilic molecules like glucose across the cell membrane. Carrier proteins are transmembrane proteins that lack a pore, when the specific large hydrophilic molecule, like glucose, binds to the specific transmembrane. The protein changes shape to allow it in without becoming a channel. 25. Carrier proteins allow much larger hydrophilic molecules through the cell membrane, still under facilitated diffusion driven by the concentration gradient, but utilizes a carrier protein, and is not a channel. 26. Channel proteins have a passageway, carrier proteins do not. 27. All active transport mechanisms require cellular energy or ATP, and transmembrane proteins. 28. Active transport mechanisms are much more selective than passive transport mechanism. 29. Active transport mechanism can allow the solute to go against the concentration gradient, or possible for a solute to move from a low concentration into a higher concentration area, inverse of passive transport mechanism. 30. There is always a transmembrane protein involved in active transport mechanisms. 31. Primary active transport. 32. One of the most important transmembrane proteins in active transport mechanism is the sodium-potassium ATPase, or the sodium-potassium pump, present across the entire eukaryotic domain. 33. The prokaryotic domain has a similar transmembrane protein to sodium-potassium pump, good evidence for a common ancestry. 34. Sodium-potassium ATPase is also an enzyme, can hydrolyze or break down ATP to produce ADP and energy. The energy is harvested by the transmembrane protein to translocate or kick out 3-sodium and bring back into the cell 2-potassium. 35. Every time the sodium-potassium ATPase hydrolyzes 1 ATP, it has enough energy to kick out 3-sodium and to bring in 2-potassium into the cell, known as primary active transport. 36. Sodium-potassium pump is a transmembrane protein and an enzyme. 37. Secondary active transport is being set up by the primary active transport. 38. Secondary active transport can transport 2 different cargoes or solutes at once. If both are transported in the same direction, it's known as co-transport or symport. 39. Secondary active transport transporting 2 solutes, but each go a different direction. It's called counter-transport or antiport or exchange. 40. Cell sets up a concentration gradient using the sodium-potassium pump. Lots of sodium outside the cell and lots of potassium inside the cell. Sodium can then get transported back into the cell by the secondary active transport mechanism. At the same time, along with glucose, by utilizing co-transport, or both go into the cell at once. 41. The co-transport step of secondary active transport, bringing in sodium and glucose at the same time, does not utilize ATP because of the concentration gradient created by the sodium-potassium pump pulling in the sodium that was expelled by the sodium-potassium pump. 42. Countertransport is driven by the concentration gradient of sodium. 43. Sodium-potassium pump helps set up the secondary active transport. 44. The cell excretes or loses protons in countertransport when it brings in the sodium from the concentration gradient. 45. Osmosis is transporting a solvent, water, across the cell membrane. Passive transport is driven by the concentration gradient of water. 46. Osmolarity is to have a way to express the concentration of particles in solution. 47. 1 molar of glucose is the same as 1 osmolar of glucose. It does not dissociate in water. 48. Some solutes dissociate in water, like sodium chloride. 1 molar becomes 2 osmolar, dissociates into Na plus and Cl minus. They are surrounded by a hydration shell of water. The Na plus is attracted to the negative on the oxygen in the water molecule, and the Cl minus is attracted to the positive charge hydrogens on the water molecules. 49. Impermeant means it is not allowed to leave. 50. The osmolarity is important to dictate the motion of water. 51. Osmotic pressure increases when osmolarity increases. 52. Water flows from a low osmotic pressure to a high osmotic pressure. Water concentration is inverse of the osmotic pressure, less water in the high osmotic pressure. Area leads to water moving to the lower water concentration or high osmotic pressure area. 53. Isotonic solution means the solution inside the cell has the same solute concentration as the outside of the cell. 54. Hypertonic solutions shrink cells, contains more salt concentration than the inside of the cell. 55. Hypertonic solutions swell cells, contain less salt concentration than the inside of the cell. 56. Vesicular transport is underactive transport, also called bulk transport. Under vesicular transport is exocytosis, when the cell needs to export something, and endocytosis, when the cell imports something. 57. There are 3 types of endocytosis. 1. Pinocytosis is known as cell drinking, when the cell brings in a large volume of water or solvent containing solutes or dissolved electrolytes. 2. Phagocytosis is known as cell eating, when the cell brings in a virus or bacterial cell. 3. Receptor-mediated endocytosis, or when hormones like insulin are brought into the cell, extremely expensive but transports extremely important cargo or chemicals. Chapter 5. Chemical messengers. 1. Chemical messengers are chemicals used by various organ systems to communicate between one another. 2. The major chemical messengers are used by 3 major organ systems: the nervous system, the endocrine system, and the immune system. 3. Chemical messengers are for communication between cells, or intercellular communication. 4. Cellular communication consists of signaling mechanisms, can rely on different types of messages, can be electrical or chemical messages. 5. Gap junctions are openings between neighboring cells, a passageway that always remains open allowing communication between the cells. 6. Chemical messengers can be classified in two ways, based on function known as functional classification, or the way they look called structural or chemical classification. 7. Signal transduction mechanism is a cascade of events allowing the cell to respond. 8. Chemical messengers can be water-soluble or fat-soluble. 9. Lipophilic means fat-soluble. 10. Direct communication is done via gap junctions between cells. 11. Connexin are the transmembrane proteins used to build gap junctions. The passageways always remain open in gap junctions. The cytosol between cells is continuous through the gap junctions. 12. Cardiac muscle fibers are cardiac muscle cells called cardiomyocytes. They have gap junctions. Action potential can spread from one cell to another in cardiac muscle fibers. Through the gap junctions, one cell activated can activate the entire organ simultaneously through the gap junctions. 13. Smooth muscle cells, especially in the digestive tract, also similarly rely on gap junctions. 14. Electrical signals are extremely fast signals. A disadvantage is electrical signaling through gap junctions have no directionality to the message, or the cell cannot control the direction of the signal. 15. Most signaling mechanisms rely on chemical signaling pathways, because it has directionality, unlike electrical signaling. 16. Indirect communication is the secretion of a chemical messenger binding to a receptor on a target cell. Chemical signaling requires a chemical messenger and a receptor to function. 17. Interaction between the chemical messenger and the receptor is extremely specific, like a lock and a key. 18. Any cell that does not display the receptor does not respond to the chemical messenger. 19. Binding between ligand and receptor is reversible because it uses weak bonds. 20. Chemical messengers need to be killed off to free up the receptor for new incoming chemical messengers. 21. Old chemical messengers are neutralized once their job is completed. 22. Chemical messenger is the same as ligand. 23. Chemical messenger or ligand changes its name depending on the organ system that uses it. The nervous system uses neurotransmitters. 24. Chemical messengers in the endocrine system are called a hormone. 25. Chemical messengers in the immune system are called a cytokine. 26. Secretory cells secrete chemical messengers. 27. Target cells receive chemical messengers using a receptor. 28. Receptors can be transmembrane proteins, located at the cell membrane where it can recognize a water-soluble chemical messenger. 29. Fat-soluble chemical messengers can go through the cell membrane. The receptor is in the target cell in the cytosol or in the nucleus. 30. Paracrines are chemical messengers that go a short distance, like growth factors, clotting factors and cytokines. 31. Not all cytokines function as paracrines. 32. Autocrines are the same chemical messengers as paracrines. However, the chemical messenger goes back to the secretory cell and can affect the secretory cell, still a short-distance chemical messenger. 33. The immune system does not exclusively rely on short-distance chemical messengers or paracrines. 34. Chemical messengers used by the nervous system are called neurotransmitters, released by presynaptic neurons. Targets are postsynaptic neurons, glands and muscles. 35. Presynaptic neurons and postsynaptic neurons. There's a microscopic space called a synapse between two neurons. The first cell is a presynaptic neuron, the second cell is a postsynaptic neuron, located after the synapse. Whereas the presynaptic neuron is located before the synapse. 36. Synapse is only a space between two neurons. 37. Neuroglandular junction is the space between the neuron and a gland. This is similar to a synapse, but the postsynaptic neuron is replaced with a gland. 38. Neuromuscular junction is the space between the neuron and a skeletal muscle fiber. The postsynaptic neuron in a synapse is replaced with a skeletal muscle fiber. 39. Chemical messenger in a synapse, neuroglandular junction and a neuromuscular junction is still a neurotransmitter. 40. Neurotransmitters are always released by a neuron. 41. Hormones are chemical messengers used by the endocrine system, are for long-distance communication. 42. Hormones are released by endocrine cells into the vascular bed, blood vessels. 43. Hormones are specific cases where chemical messengers have to travel long distances to reach the target, like insulin or ADH, released by the hypothalamus, stored by the posterior pituitary gland. 44. ADH is also known as vasopressin. 45. Hypothalamus is a neural structure producing chemical messengers that function as a hormone. The hormone, ADH, is known as a neurohormone. 46. 99% of pancreatic cells have digestive functions. Only 1% of pancreatic cells have endocrine functions. The beta pancreatic cells produce and release insulin. The alpha pancreatic cells produce and release glucagon. 47. Insulin is involved in maintaining the concentration of glucose in the blood plasma. 48. Vasopressin or ADH antidiuretic hormone, while dehydrated ADH is released inducing the kidneys to conserve water, urinate much less, urine turns yellow. 49. Table 5.1 through 5.6 add to notes. 5.1 and 5.2 are in the lecture slides. 5.3 to 5.6 are in the book PG 141. 50. Lipophobic means hydrophilic or water-soluble. 51. Lipophilic means fat-soluble. 52. Water-soluble chemical messenger receptors have to be on the plasma membrane. 53. Chemical messengers that are fat-soluble go straight through the membrane to the receptor in the cytosol or the nucleoplasm through the nuclear envelope. 54. Hormones released into the blood are released into the vascular beds. 55. Chemical messengers can be simple or complicated chemicals like macromolecules or proteins. 56. Peptides or proteins can be used as a chemical messenger. 57. 1 to 99 amino acid sequence is known as a peptide. If there's 100 or more amino acids in a sequence, it will be called a protein. 58. A protein means the same thing as a polypeptide. 59. Peptides are produced as an inactive form, from the prehormone to the prohormone to the functional hormone. 60. Specific enzymes like protease can cut off some amino acids along the chemical messenger to release a functional hormone from the pre or prohormone. 61. The parathyroid hormone is very important, involved in maintaining calcium homeostasis. Parathyroid hormone produced by the parathyroid gland, located posteriorly to the thyroid gland. There are 4 to 8 parathyroid glands per human being. 62. Parathyroid hormone is known as PTH, released when calcium concentration in the blood drops. 63. Parathyroid hormone is released as a precursor, is cleaved to release a functional hormone. 64. Since the hormone protein is released, it has to be translated by ribosomes associated with the endoplasmic reticulum and shipped to the Golgi apparatus to be exported out of the cell through exocytosis, an active transport mechanism. The peptide has a signal peptide to tell the Golgi apparatus where it needs to go. 65. Parathyroid gland is in the endocrine system, controlling the body to respond to a drop of calcium in the blood. 66. Fat-soluble chemical messengers go through every single physical barrier at the cellular level, combined to receptors in the cell. The hormone receptor complex acts as a transcription factor. 67. Transcription factor can increase or shut down transcription. 68. DNA can be used to produce messenger RNA through transcription. The transcription factor can affect the transcription step. The messenger RNA being produced leads to protein synthesis. So the transcription factor can change if the protein is produced faster or at all. Any fat-soluble chemical messenger is potentially dangerous, can shut down protein synthesis. 69. Some toxins are fat-soluble. Shutting down transcription shuts down protein production. 70. HRE hormone response element acts as a transcription factor, recognizes a specific sequence on the DNA molecule or a gene that codes for a protein. 71. Humans have around 20,000 genes, but there are over 100,000 different proteins in the cells. One gene can generate multiple versions of a protein. 72. Ligands or messengers or hormones are fat-soluble chemicals. 73. Water-soluble messengers remain outside the cell, cannot go through the cell membrane. Message has to be conveyed through a transmembrane protein. When the chemical messenger binds to the transmembrane protein, it opens the pore to allow movement through it. 74. Things coming into the cell through the transmembrane proteins are called influx. things leaving is called efflux, passive transport through concentration gradient known as facilitated diffusion. 75. Sodium, chloride, and calcium all influx into the cell. 76. Potassium is effluxed out of the cell through the transmembrane proteins. 77. Water-soluble chemical messengers bind to the outside of the transmembrane proteins to allow it to flux or open. The secondary messengers like sodium, chloride, and calcium get in the cell. The water-soluble chemical messengers stay outside the cell. 78. The cell can change through electrical signaling from the secondary chemical messengers, can induce contraction if the cell is a muscle cell, can induce secretion if the cell is part of a gland. Or if it's a neuron, it releases neurotransmitters, can activate a passage leading to smooth muscle contraction. 79. 3 types of muscle are smooth, skeletal and cardiac. 80. Enzyme-linked receptors are transmembrane protein receptors that are also an enzyme, acts as a receptor but is also an enzyme. 81. Kinase means the ability to add a phosphate group onto another protein. Tyrosine kinase can phosphorylate a specific amino acid called tyrosine onto a protein. Any protein that has a tyrosine will have its tyrosine phosphorylated. 82. Tyrosine is a specific amino acid, one of the 20 amino acids. 83. Tyrosine kinase is an enzyme-linked receptor and a transmembrane protein, does not have a pore. 84. Insulin can act as a hormone on the target cells, through the enzyme-linked receptors. 85. A major family of receptors is called G-protein-linked receptors. They lack a pore. The messenger is a water-soluble chemical messenger, remains outside the cell, binding to the receptor. 86. The G protein has 3 subunits. The alpha subunit is the focus. There is the beta and gamma, but not the focus here. 87. Once the water-soluble messenger binds to the transmembrane protein, the transmembrane protein is linked to a G protein. The receptor is activated, activating the G protein, specifically the alpha subunit, goes on to activate an ion channel, changing the electrical properties of the cell. 88. The alpha subunit of the G protein can activate another transmembrane protein that's also an enzyme. The adenylate cyclase can convert ATP into cyclic AMP or CAMP, acts like a second messenger activating a protein kinase enzyme. Can add phosphate groups onto another protein. 89. Protein kinase phosphorylates a target protein. Adding a phosphate to a substrate, the protein becomes activated and cascades, leading the protein to respond. 90. Page 143, Figure 5.19, Signal amplification. The second cyclic AMP can amplify the response. Started with one chemical messenger, can produce hundreds of thousands of signaling cascades. 91. G-protein linked receptors can also be called G-protein coupled receptors. Chapter 6. Endocrine system. 1. The endocrine system controls all remaining organ systems, controls 11 other organ systems at a time, except the nervous system. 2. The endocrine system relies on chemical messenger hormones to communicate with all other organ systems to induce them to carry out functions dictated by the endocrine system. 3. The hypothalamus is the master switch of the endocrine system, controls all other endocrine structures, but belongs to the nervous system. 4. The anterior lobe of the pituitary gland is part of the endocrine system along with the posterior lobe. 5. The endocrine system has endocrine reflexes like the nervous system. 6. There are pure endocrine organs. They only produce and release hormones: single pituitary gland, single pineal gland, one thyroid gland, the parathyroid glands, 4 to 8 of them, and 2 adrenal glands sitting above each kidney. 7. The thyroid gland is anterior, and the parathyroid glands are located posterior to the thyroid gland. 8. Superficial adrenal cortex and a deeper adrenal medulla producing and releasing different types of hormones under different scenarios. 9. Neuroendocrine organ is the hypothalamus. 10. There are other organs that produce and secrete hormones that are not in the endocrine system. 11. The thymus is located anterior and superior to the heart, is a lymphatic organ that produces hormones. 12. The pancreas is a digestive organ. 99% of pancreatic cells are involved in producing and secreting digestive juice or pancreatic juice. Only 1% of all pancreatic cells have endocrine functionality, or producing and secreting hormones. 13. The pancreas relies on a negative feedback mechanism to control the blood concentration of glucose. 14. Gonads, testes in males and ovaries in females, are part of the reproductive system, also has endocrine functionality, producing and releasing hormones into the bloodstream. 15. The heart is part of the cardiovascular system, but also secretes hormones into the bloodstream. 16. The stomach, liver, and small intestine are organs of the digestive system, also secretes hormones into the bloodstream. 17. The kidneys are part of the urinary system, the kidneys secrete hormones into the bloodstream. 18. The placenta also produces and releases hormones, is the only organ in the human species that contains tissues coming from two distinct individuals. The mother's immune system does not reject the tissue, despite it coming from the fetus, even though it's a distinct human being. 19. 2 months after the embryonic development period, the placenta takes over for fetal development. 20. The hypothalamus controls the entire endocrine system by keeping a leash on 3 targets: the anterior pituitary gland. The posterior pituitary is used as a storage site storing 2 hormones produced by the hypothalamus, and the adrenal medulla, or the deeper region within each adrenal gland. 21. The hypothalamus belongs to the nervous system, but controls the endocrine system. 22. Chemical messengers released by the hypothalamus are called regulatory factors. 23. 2 types of regulatory factors are the releasing hormones and the inhibiting hormones. 24. Corticotropin or CRH is a releasing hormone released into the bloodstream and gets in the anterior lobe of the pituitary gland. 25. The vascular bed connecting the hypothalamus to the anterior pituitary gland is unique, it's called the hypophyseal portal system. 26. Hypophyseal portal system means there's a capillary bed in the hypothalamus, traveling to the second capillary bed in the anterior lobe of the pituitary gland. The blood vessel connecting the 2 capillary beds is called a portal vein. 27. Every portal system consists of back-to-back capillary beds. 28. The hypophyseal portal system allows the hypothalamus to control the release of hormones going to the anterior lobe of the pituitary gland. 29. Once the anterior pituitary gland receives corticotropin or CRH from the hypothalamus through the hypophyseal portal system, it will produce and secrete ACTH or adrenocorticotropic hormone, targeting the adrenal cortex, or the superficial layer in the adrenal gland. 30. ACTH hitting the adrenal cortex induces the adrenal cortex to release steroid hormones or fat-soluble hormones. They go straight through the cell membrane and don't require a transmembrane protein. Receptors are in the cytosol or nucleus of each cell. 31. Thyrotropin or TRH is released by the hypothalamus, is a releasing hormone. Upon reaching the pituitary gland, it will induce the release of the thyroid-stimulating hormone known as TSH. 32. Gonadotropin releasing hormone or GnRH from the hypothalamus induces the anterior pituitary gland to release 2 hormones. These are the luteinizing hormone or LH and the follicle-stimulating hormone or FSH. 33. Prolactin-releasing hormone or PRH from the hypothalamus induces the anterior pituitary gland to release prolactin or PRL, induces the mammary gland to produce milk for lactating mothers. 34. Growth hormone-releasing hormone or GHRH from the hypothalamus induces the anterior pituitary gland to release growth hormone or GH. 35. There are neurosecretory cells in the hypothalamus. 36. The infundibulum is the stalk connecting the hypothalamus to the pituitary gland. 37. Deep in the infundibulum is the first capillary bed. In the anterior pituitary gland is the second capillary bed, part of the hypophyseal portal system connecting the hypothalamus to the anterior pituitary gland. 38. Within the hepatic portal system, the first capillary bed is perfusing the jejunum part of the small intestine, the second capillary bed would be perfusing the liver. The portal vein would absorb nutrients from the small intestine in the jejunum region, and bring it to the liver for detoxification purposes. 39. Neurosecretory cells in the hypothalamus are actually neurons. They are producing chemical messengers into the bloodstream, no longer called neurotransmitters. They are called regulatory factors or hormones since they're secreted into the bloodstream. 40. Neurotransmitters are chemical messengers that are close distance between neurons. 41. Regulatory factors that affect the anterior pituitary gland are neurosecretory cells. 42. Posterior pituitary gland only accept two hormones produced by the hypothalamus, stored in the posterior pituitary gland, are ADH or vasopressin and oxytocin. 43. Oxytocin is produced by neurons located in the supraoptic nucleus. Supra means above, optic means optic nerve. 44. ADH or vasopressin is produced by neurons located in the nucleus called paraventricular nucleus next to the third ventricle. Figure 6.3 page 151 of textbook. 45. Figure 6.5 page 152 shows the hypothalamic tropic hormones or regulatory factors, either releasing or inhibiting hormones. 46. Regulatory factors known as inhibiting hormones. If the hypothalamus sends the prolactin inhibiting hormone, the production of prolactin is inhibited. 47. If the hypothalamus sends the growth hormone inhibiting hormone, the anterior pituitary gland shuts down the production and release of the growth hormone. 48. Regulatory factors either induce or inhibit the anterior pituitary gland to produce and release hormones. 49. Hypophyseal portal system is the same as the hypothalamic pituitary portal system. 50. Prolactin inhibiting hormone induces the anterior pituitary gland to stop producing prolactin. 51. Cortisol is a specific example of steroid hormones or fat-soluble hormones. 52. Growth hormone targets the liver and cells throughout the body for cell growth, induces cell division. 53. The adrenal medulla is the deeper regions inside each adrenal gland. 54. The adrenal medulla secretes chemical messengers acting both as a neurotransmitter or hormone depending on which cell secretes them, whether in a synapse, a neuromuscular or neuroglandular junction. If they are released into the bloodstream, then they act as hormones. 55. Adrenaline and noradrenaline are classified under a larger term called catecholamines. Adrenaline is also called epinephrine and noradrenaline is called norepinephrine. 56. The adrenal medulla produces and secretes 80% of catecholamines as adrenaline, 20% is noradrenaline. 57. The cells producing and secreting epinephrine and norepinephrine are called modified ganglia. They are not ganglionic neurons and instead are called chromaffin cells. 58. Release of adrenaline and noradrenaline in stressful situations induces the person to breathe faster, to take deeper breaths. Dilates the blood vessels perfusing the skeletal muscles, allowing the skeletal muscles to receive more oxygen and nutrients and dispose of waste quicker. 59. Epinephrine is a water-soluble molecule, not a steroid or fat-soluble. 60. The adrenal cortex is the superficial layer of the adrenal medulla. 61. The adrenal cortices release steroid or fat-soluble hormones, known as adrenocortical hormones. 62. Three families of steroid hormones are mineralocorticoids involved in maintaining homeostasis of salts, most importantly sodium, glucocorticoids maintain homeostasis of blood glucose, and gonadocorticoids producing DHEA. 63. Aldosterone is the most important hormone of mineralocorticoids. 64. Cortisol or hydrocortisone is the most important hormone of glucocorticoids. 65. DHEA or dihydropyandrosterone is a major androgen, male sex hormone, can be converted to a female hormone called estrogen, is the most important hormone of gonadocorticoids. 66. Estrogen protects women during menopause, protecting the heart and bones. When younger, estrogen is produced by ovaries. After menopause, the ovaries no longer works, compromised of estrogen, has to be extracted from the adrenal cortices producing DHEA converted to estrogen. 67. Some cells spend half of all cellular energy to maintain sodium homeostasis. Where salt goes, water follows due to osmotic pressure. 68. Once adrenal cortices release steroidal hormones, under long-term stress. 69. The adrenal medulla releasing adrenaline and noradrenaline is under short-term stress, or fight or flight scenario. 70. The three different layers in the adrenal cortex are the zona glomerulosa, superficial, the zona fasciculata, middle layer, and the zona reticularis, deepest layer. 71. The zona glomerulosa produces mineralocorticoids, steroid hormones involved in maintaining electrolyte homeostasis. 72. The zona fasciculata produces glucocorticoids and gonadocorticoids. 73. The zona reticularis produces gonadocorticoids and glucocorticoids. 74. Hormonal concentration is talking about the chemical messenger, also requires a receptor on the target cell. 75. Factors can affect a response to a hormone, like the concentration of the chemical messenger, how many receptors the target cell displays known as receptor density, and the influence by other hormones. 76. When hormones arrive at the target cells, often there are other chemical messengers there simultaneously, 3 types of influences by other hormones. They can oppose each other with antagonistic effects, they can help each other with synergistic effects. Or one hormone function is allowed by the presence of another hormone known as permissiveness effect. 77. Antagonistic effect example is PTH and calcitonin. PTH increases the concentration of calcium in the blood back to normal levels. Calcitonin will decrease the blood calcium back to normal levels. 78. Insulin will decrease glucose concentration in the blood. Glucagon will increase glucose concentration in the blood. 79. Epinephrine hormone increase the heart rate and so does norepinephrine. Both simultaneously, the effect of both on the heart increases the heart rate by more than both combined, known as synergistic effect. 80. Permissiveness effect example is epinephrine dilates the bronchioles smooth muscle, works efficiently with the presence of thyroid hormones. 81. Growth hormones without thyroid hormones will make the limbs grow disproportionately. The thyroid hormones make sure the limbs grow proportionately, also a permissiveness effect. 82. There are 3 types of endocrine reflex: neural stimulus, hormonal stimulus and humoral stimulus. 83. The neural stimulus is during short-term stress scenarios or fight or flight situations. The hypothalamus sends out neurons called preganglionic motor fibers. They are axons wrapped in a nerve called the splanchnic nerve going down to the adrenal medulla. 84. There are no postganglionic neurons in the adrenal medulla. There is instead chromaffin cells. They are modified ganglionic cells. 85. Adrenaline and noradrenaline also increase the contractility of the heart or the strength of the heart. 86. Long-term stress is under the hormonal stimulus endocrine reflex, under the hypothalamus through the hypophyseal portal system. 87. The hypothalamus releases CRH or corticotropin-releasing hormone targeting the anterior pituitary gland. The anterior pituitary releases ACTH or adrenocorticotropic hormone. ACTH uses the bloodstream to go to the adrenal cortices. 88. Adrenocortical hormones are essential for life. If they're damaged, humans can only live for 24 to 48 hours without them. 89. Physical trauma can induce long-term stress, as well as any illness. 90. The humoral stimulus is activated by electrolytes or simple chemicals like potassium, calcium or glucose. 91. When blood glucose increases, for example after a meal, the beta cells of the pancreas detects the spikes in blood glucose, producing and secreting insulin into the bloodstream, which goes throughout the body to induce the cells to absorb glucose. 92. When cells throughout the body pick up glucose, blood glucose concentration drops. 93. When blood glucose goes to a normal level, the beta cells of the pancreas stop releasing insulin. 94. Insulin is a protein used as a chemical messenger. 95. When blood glucose decreases, detected by another type of cells in the pancreas, called the alpha cells of the pancreas, they release glucagon, another protein acting as a hormone. Glucagon goes to the liver to induce hepatocytes or liver cells to proceed to break down glycogen stores to release glucose back into the bloodstream to increase glucose. 96. When glucagon induces hepatocytes to break down glycogen, it's called glycolysis. 97. Glycogen is a polysaccharide, being hydrolyzed to release glucose back into the bloodstream. 98. When blood potassium increases, the adrenal cortex releases a steroid hormone called aldosterone, inducing the kidney to expel potassium into the urine, dropping blood concentration of potassium. When potassium levels are back to normal, aldosterone is no longer produced. 99. When blood calcium is low, the chief cells of the parathyroid glands release parathyroid hormones. The parathyroid hormones have 3 functions: inducing bone resorption or activating osteoclasts to release calcium, also decreases calcium lost through urinary output. Also activates vitamin D into calcitriol. Activated form of vitamin D, calcitriol, can increase calcium absorption through the digestive tract. 100. Calcium blood levels increasing only applies under two cases: during pregnancies, or when a child is actively growing. Non-pregnant adults not growing do not have this condition. 101. Calcitonin has the opposite effect during pregnancy or when a child is growing, induces osteoclasts to store calcium in the bones. 102. 99% of calcium is stored in the bones, used as a storage bank for calcium. Chapter 7. Nerve cells and electrical signaling. 1. The capability of generating electrical signals. The cell in biology cannot generate electrons like in electrical wires, the cell would die. 2. Resting membrane potential and how do neurons generate, establish, and maintain the resting membrane potential, concepts for this lecture. 3. Electrical signaling is possible just by changing a membrane potential. 4. Specific example of utilizing membrane potential and using it as an electrical signal is undergraded potentials. 5. Nervous system is divided into central nervous system and peripheral nervous system. Central nervous system includes the brain and spinal cord only. Everything else in the nervous system not physically a part of the brain or spinal cord is part of the peripheral nervous system. 6. Information that goes toward the central nervous system is called afferent, sensory, or ascending, all mean information being conveyed to the central nervous system, from the spinal cord to the brain. 7. Efferent pathway transmits information away from the central nervous system, also called descending pathway or motor pathway, transmits motor decision, originate from the brain or the spinal cord. 8. The sensory pathways rely on senses, sensing a stimulus and sending it to the central nervous system. 9. Somatic or autonomic organs receive the information from the central nervous system. 10. The four somatic general sense are sense of touch, temperature, pressure, and pain. 11. The 5 special sense are sense of sight, smell, taste, hearing, and balance. 12. The visceral sense cannot be told the exact location, like where discomfort originates. 13. Discomfort in visceral organs cannot be exactly pinpointed. 14. Somatic means voluntary control, autonomic means involuntary control, includes the sympathetic and parasympathetic motor outputs. 15. Under sympathetic motor outputs, there's cardiac muscle, smooth muscle, glands, and adipose tissue. 16. Parasympathetic system targets the enteric nervous system, specifically the digestive system. 17. The autonomic division includes the sympathetic, parasympathetic, and the enteric nervous system. 18. Sympathetic system preps the body for fight or flight situations. 19. Parasympathetic system is the inverse of the sympathetic, the rest and digest situations. 20. Sympathetic and parasympathetic divisions do not work together, they suppress each other, only time they are both activated is during sexual intercourse. 21. Neurons can be classified using 2 classification methods, classified based on structure or the way they look, or classified based on functionality. 22. 4 families of structural classification for neurons. Anaxonic neurons lack an axon, bipolar neurons have a dendrite and axon separated by the cell body. The pseudo-unipolar neuron has the cell body on the side, making the axon continuous from one side to the other. The multipolar neuron is a prototype motor neuron, thousands of dendrites on one side, one axon on the other side. 23. Central or peripheral neurons are classified based on the cell body they have. If the cell body is located in the central nervous system, it's classified as a central neuron. If the cell body is located physically outside the brain and spinal cord, it's classified as a peripheral neuron. 24. The pseudopolar neuron has a cell body outside the central nervous system, it's a peripheral neuron. 25. The cell membrane of neurons can be used to store potential energy. The cell membrane is capable of separating charges in a neuron, even though cells are neutral themselves. Only at the membrane, the outer and inner membranes called the outer and inner leaflets, is there a charge. 26. The cell membrane by splitting opposite charges from one another stores potential energy, the charges coming back together releases energy. 27. On the cell membrane there's transmembrane proteins acting as channels known as leak channels, can leak potassium or sodium. 28. 2 specific types of transmembrane proteins, one leaks potassium and one links sodium. They are cation channels with a pore or passageway which remains open constantly, located at the cell membrane or phospholipid bilayer. 29. There are more leak potassium channels open than leak sodium channels open, or the cell allows 25 times more potassium to leave the cell than letting in sodium. The cell loses positive charge faster than it gains positive charge. The cell becomes less positive than the outside of the cell. 30. The chemical gradient drives the potassium efflux and sodium influx. 31. The lessened positivity inside the cell will slow down the potassium efflux and increases the sodium influx, because the electrical force will oppose the movement of positive charges from leaving the cell. 32. The surface of the inner leaflet is negative charge, the outer membrane of the leaflet is positively charged, the cytosol is neutral, the stored energy is in the different charges on the leaflets. 33. Equilibrium for the cell is negative 70 mV. 34. Each ionic species has its own equilibrium potential. Sodium has plus 60 mV, potassium has negative 94 mV. Since the cell membrane is more permeable to potassium efflux, the cell membrane potential at equilibrium is closer in millivolts to potassium. 35. The membrane potential of negative 70 mV would disappear over time. The concentration for potassium and sodium would become equal inside and out. 36. The cell uses cellular energy to make sure the negative 70 mV remains the same by reintroducing sodium-potassium ATPase, utilizing primary active transport to kick out 3 sodium and bring in 2 potassium and use 1 ATP, establishes and maintains the resting membrane potential of neurons. 37. The resting membrane potential can fluctuate anywhere from negative 5 mV to negative 100 mV depending on the cell. Neurons specifically have negative 70 mV. 38. Every single cell in the body has a resting membrane potential. 39. Inner leaflet of plasma membrane is less positively charged. The outer leaflet of the plasma membrane is positively charged. 40. Membrane potential energy is expressed in volts. 41. If the resting membrane potential value is changed, it can be utilized as a signal. 42. There are 2 types of electrical signals, the action potential and the graded potential. 43. There are 3 types of graded potential, the receptor, generator, and the postsynaptic potential. 44. Graded potentials are used for short-distance communication. The graded potential loses signal intensity proportionately to the distance being covered. 45. The axon hillock is the most important location of the neuron. It's the central processing unit of the neuron, can decide whether or not to fire an action potential. 46. Postsynaptic potential needs to get to the axon hillock, but distance greatly lowers the intensity of the signal. 47. Short-distance propagation is the graded potential. 48. Conduction means electrons running along a surface like a wire, but in biology those free radicals are high-energy electrons, which kill cells. Conduction is not proper terminology, instead use propagation. 49. Action potential signals can be used for short or long distance. The signal amplitude does not lower with distance, retains 100% of its energy at its destination. 50. Action potential is difficult to generate, can only be generated at 100% intensity every time, all or none principle. 51. Action potentials propagating through a neuron is propagated by positive feedback mechanisms. 52. Action potentials cannot be added on top of one another. The neuron needs to wait for it to stop before it can be generated again. There cannot be multiple action potentials simultaneously. 53. Graded potentials can be added on top of one another to prevent the dissipation of signal intensity due to distance. 54. Postsynaptic potentials also have excitatory and inhibitory postsynaptic potentials. 55. Postsynaptic potentials are induced by the presynaptic neurons for the postsynaptic neurons to produce postsynaptic potentials. 56. Excitatory postsynaptic potentials. There are neurotransmitter channels on the postsynaptic neuron that depolarize the resting membrane potential, either the dendrite or the soma, the cell body excluding the axon hillock. These channels are transmembrane proteins called neurotransmitter-gated cation channels activated by neurotransmitters released by the presynaptic neuron. 57. At rest, the inner leaflet of the neuron is negative. Depolarization makes the inner leaflet positive and the outer leaflet negative. 58. Neurotransmitters activate transmembrane proteins. They flux anions like chloride in inhibitory postsynaptic potentials, makes the inner leaflet even more negative than before. inhibiting the postsynaptic neuron from generating an action potential, hyperpolarizing the membrane. 59. Axon hillock, in order to fire an action potential, needs the inner leaflet to be depolarized or more positive, cannot fire an action potential when hyperpolarized or made even more negative. 60. Neurotransmitters are released by presynaptic neurons, chemical messengers used by neurons in the nervous system. 61. EPSP stands for excitatory postsynaptic potentials. 62. IPSP stands for inhibitory postsynaptic potentials. 63. If the postsynaptic neuron receives the neurotransmitters glutamate, one of the 20 amino acids used to build proteins, being used as a chemical messenger here, acetylcholine or ARC, serotonin, and ATP all are used here as neurotransmitters. 64. ATP-gated ion channels involved in pain perception, cloned a brand new ATP-gated ion channel in the 90s for Dr. La's PhD thesis, not relevant to lecture material. 65. When glutamate, ARC, serotonin and ATP bind to the neurotransmitter-gated cation channels, they are produced and released by the presynaptic neuron. They go through the synapse to the postsynaptic neuron through simple diffusion. On the plasma membrane on the dendrites are the cation channels where the neurotransmitters bind and open the channels, sodium and calcium influx, potassium efflux, much more influx than efflux, inner membrane become depolarized flipping the polarity. 66. EPSPs on the plasma membrane of the dendrites and cell body of the postsynaptic neurons is the formation of the EPSPs that try to get to the axon hillock of the postsynaptic neuron. 67. Inhibitory neurotransmitters called GABA and glycine, one of the 20 amino acids, most simple amino acid and lacks a chiral center. 68. GABA is released by neurons in the brain. 69. Glycine is released by neurons in the spinal cord. 70. Both GABA and glycine are produced and released by presynaptic neurons released into the synapse or synaptic cleft by simple diffusion, leaving the presynaptic neuron binding to the receptor on the cell membrane of the postsynaptic neuron. When they bind, they open the pore, chloride influxes into the neuron, the membrane becomes hyperpolarized, making the inner leaflet more negative, creating IPSPs or inhibitory postsynaptic potentials, preventing the axon from generating action potentials. 71. The threshold potential has to be reached before the axon hillock can fire an action potential. The threshold potential is a more positive charge than the equilibrium charge of the neuron at negative 70 millivolts. 72. Graded potentials become generated and die out on the millivolt graph. However, multiple EPSPs can break through the threshold potential and fire an action potential. Chapter 7 Part 2, Nerve Cells and Electrical Signaling. 1. Action potentials are the most important electrical signaling mechanism, allowing neurons to send out signals. 2. Action potentials can only be generated at a very specific point in a neuron and only propagated to another specific area of the neuron. 3. Action potentials are electrical signals that can be sent out in a single direction. 4. All graded potentials, EPSPs and IPSPs, are generated only at the dendrite or at the cell body of the postsynaptic neuron. The cell body excludes the axon hillock in this context. 5. EPSPs and IPSPs will lose their intensity the farther they have to travel, but they can be added on top of one another to increase their intensity. 6. The net sum of all the EPSPs and IPSPs have to reach the threshold potential at the axon hillock to generate an action potential. 7. Threshold potential is negative 55 millivolts or more positive to generate an action potential. 8. Axon hillock adds up all EPSPs and IPSPs electrical charge to see if it reaches the threshold potential. 9. EPSPs are depolarization signals, making the inner leaflet more positive, flipping the polarity of the two leaflets of the cell membrane. 10. IPSPs are hyperpolarization signals, making the inner leaflet more negative. 11. AP means action potential. 12. Action potential consists of three distinct phases called phases 1 through 3. 13. Phase 1 of the action potential is called a depolarization phase. The threshold potential of negative 55 millivolts was met from the resting membrane potential of negative 70 millivolts. 14. The depolarization phase or phase 1 is mediated by openings of ion channels. These ion channels are transmembrane proteins acting as a receptor that is sensitive to voltage change across the cell membrane. When they open, they flux ionic species. Specifically influx only sodium cations at the axon hillock of the neurons. 15. Sodium massively accumulates on the inner leaflet of the axon and becomes more positive, moving from a negative value to a positive value, due to a net gain of positive charge, from the massive influx of sodium cations. 16. Sodium cations are fluxing through voltage-sensitive sodium channels in the depolarization phase or phase 1 of the action potential. 17. Phase 2 of the action potential, the membrane potential goes down back to a negative value. Phase 2 is called the repolarization phase, due to the activation of another voltage-sensitive ion channel, opens and only fluxes potassium. The flux is efflux, or leaving the axon hillock. The axon hillock loses potassium cations and losing positive charge, the potential becomes negative. 18. Phase 3 of the action potential is mediated by the same potassium efflux. The voltage-sensitive potassium channel in phase 2 remains open longer than the sodium channels in phase 1. There's more potassium leaving the cell, making the inner leaflet even more negative than when at rest, known as hyperpolarization. 19. Phase 3 is when the neuron returns to the resting membrane potential of negative 70 mV. 20. Axon hillock cannot fire multiple action potentials within a span of time called an absolute refractory period, contains the entirety of phase 1 of the action potential. 21. Phase 2 of the action potential. If the signal is strong enough, the axon hillock can fire an action potential. This is known as the relative refractory period. 22. The birthplace of all action potentials occurs at the axon hillock, part of the cell body of the neuron. 23. The axon hillock connects the cell body to the axon in a neuron. 24. The generation of an action potential occurs at the axon hillock. The propagation of action potentials occurs along axons. 25. Electrons are not involved in action potential generation. It's about changing the polarity across the inner and outer leaflet of the membrane by changing the movement of ionic species, specifically sodium cations and potassium cations. 26. Conduction will be a trick question. Do not use conduction. 27. Continuous propagation of action potential continues along unmyelinated axons, or axons without multiple layers of myelin sheathing around their structure. 28. Continuous propagation also occurs along muscle fibers, skeletal muscle fiber, cardiac muscle fiber, and smooth muscle cells. 29. The continuous propagation mechanism is very slow. Without myelin sheathing, there is a loss of charge to the extracellular fluid. The cell membrane is not insulating enough. As the potential moves along the axon, the action potential is losing signal intensity. 30. Continuous propagation moves at walking pace or relatively slow, 1 to 2 miles per hour, due to lack of insulation from lacking myelin sheathing. 31. Voltage-sensitive sodium channel allows massive sodium influx. The channel closes quickly by a mechanism called ball and chain. Part of the channel that occludes the pore is a ball, so sodium cannot get in. This is why phase 1 of the action potential is brief. 32. The voltage-sensitive potassium channel does not have the ball and chain, can remain open longer, repolarizes and then hyperpolarizes the membrane. Phase 2 and 3 is due to the opening of the voltage-sensitive potassium channel. 33. Action potentials can only move forward due to the absolute refractory period. 34. Each site can only activate each forward site because of the absolute refractory period acting as a cooldown. A one-way mechanism leaving the axon hillock, moving along the axon and hitting the axon terminal at the distal end of the axon. 35. Saltatory propagation occurs only in myelinated axons, or axons that have myelin sheathing wrapping around them up to 50 times, insulates the axon and prevents charge loss when the axon is generating and propagating an action potential. 36. There are regions in an axon with no myelination. They are called nodes of Ranvier. They are responsible for where action potentials are created. When there is myelination, action potentials cannot be created, so nodes of Ranvier are necessary for their creation. 37. Saltatory means jumping. 38. Action potentials appear to jump from one node of Ranvier to the next. 39. Action potentials are generated at each node of Ranvier, but then it dies at the same node of Ranvier, inducing the next node of Ranvier to produce the next action potential, creates a jumping effect. The absolute refractory period prevents the action potentials from traveling backwards. 40. The speed of action potential propagation depends on two factors: the diameter of the axon. The larger the axon, the faster the propagation, and the extent of myelination. The more myelination, the faster an action potential can travel. Can go up to 300 miles per hour in saltatory propagation compared to 1 to 2 miles per hour under continuous propagation. 41. Saltatory propagation relies on a positive feedback mechanism. 42. Voltage-sensitive sodium channels are necessary for the generation of an action potential. 43. The action potential dies when it reaches the axon terminal. The plasma membrane of the axon terminal does not have voltage-sensitive sodium channels located at the cell membrane. 44. The action potential remains at the same intensity throughout the neuron. Each node of Ranvier generates its own action potential. 45. There are two types of membrane potentials: graded potentials and action potentials. 46. Graded potentials can only be generated at the dendrites or at the cell body, excluding the axon hillock. 47. Graded potentials are specifically EPSPs and IPSPs. They have to move and reach the axon hillock. 48. The cell membrane of the axon hillock needs to reach the threshold potential to generate an action potential. 49. The action potential reaches the axon terminal and dies. It induces the neurotransmitter to be released. 50. The axon terminal contains voltage-sensitive calcium channels. When the action potential reaches the axon terminal, it depolarizes the membrane. The voltage-sensitive calcium channels are activated. They open, allowing a massive calcium influx into the axon terminal. The calcium induces vesicles, little bags containing neurotransmitters, diffuse with the cell membrane of the axon terminal, dumping the neurotransmitters in the synapse. Moving on through simple diffusion and binding to receptors on the dendrites of the postsynaptic neuron to generate EPSPs or IPSPs depending on the type of neurotransmitter. 51. Excitatory neurotransmitters like glutamate, acetylcholine, ATP, or serotonin bind to the respective receptors on the dendrites or the cell body of the postsynaptic neuron, then generate EPSPs, which help each other to reach the axon hillock to reach threshold potential to fire another action potential. 52. Inhibitory neurotransmitters like GABA or glycine induce the dendrites or cell body of the postsynaptic neuron to generate IPSPs, hyperpolarizing the membrane, lessening the chance of the axon hillock of generating an action potential. 53. Each neuron in the brain makes synaptic contact with 10,000 other neurons. 54. When EPSP and IPSP neurotransmitters bind to the receptors, they are called neurotransmitter-gated ion channels. 55. When excitatory neurotransmitters open, they flux multiple ionic species like sodium, calcium, and potassium through the same pore. 56. IPSPs flux only negatively charged chloride anions. Chapter 8. Synaptic Transmission and Neural Integration. 1. When chemical messengers migrate through the synapse and bind to receptors on the postsynaptic neuron dendrite, this is known as synaptic transmission, requires communication between at least two neurons. 2. The nervous system is the most complex organ system and gives rise to thought, but the nervous system has only three functions. It has sensory, motor, and integration functions. Sensory allows it to receive information. Motor is a reflection of the ability to send out a decision. Integration is digesting sensory information. 3. A single cell is capable of performing all three of the fundamental functions of the nervous system. Neurons can receive information. Integration occurs at the axon hillock. And the motor decision is the action potential propagating down the axon and releasing its neurotransmitter. 4. Synaptic transmission involves presynaptic and postsynaptic neurons. 5. The receptor is physically located on the dendrite of the postsynaptic neuron or the cell body, excluding the axon hillock. 6. There are excitatory and inhibitory neurotransmitters. Excitatory neurotransmitters are acetylcholine, ATP, glutamate, and serotonin. Inhibitory neurotransmitters are GABA and glycine. 7. Synaptic transmission requires at least two neurons. 8. Electrical synapse is about the two neurons physically contacting each other through gap junctions, which are always open between two neighboring cells. 9. Synapse is a microscopic space between two neurons. 10. Electrical synapses have no directionality, but communication is extremely fast. 11. Chemical synapse is the majority of cell-to-cell communication. 12. Advantage of chemical synapse over electrical synapse is directionality. However, chemical synapse is slower. Chemicals are called neurotransmitters. 13. Presynaptic neuron is the neuron before the synapse. Postsynaptic neuron is the neuron after the synapse. 14. Synapse is synonymous with synaptic cleft. 15. Synaptic vesicles contain the neurotransmitters. 16. The action potential arrives at the synapse, depolarizing the membrane. This is picked up by a voltage-sensitive calcium channel, leading to a massive influx of calcium. This influx induces the synaptic vesicle to fuse with the cell membrane of the axon terminal, allowing the synaptic vesicle to dump the neurotransmitters into the synapse. Neurotransmitters are passively transported across the synaptic cleft through simple diffusion. Eventually binds to the receptor on the cell membrane of the postsynaptic neuron. 17. The receptor on the postsynaptic neuron is a dendrite or a soma. Soma means cell body. This excludes the axon hillock. 18. The dendrite or soma will then generate EPSPs or IPSPs depending on the type of neurotransmitter that binds to the receptor. 19. The neurotransmitter binding to the receptor on the cell membrane of the postsynaptic neuron is very specific, like how acetylcholine binds only to the acetylcholine receptor. 20. Ligand-gated or neurotransmitter-gated or chemical-gated channels are the receptors on the dendrite or soma of the postsynaptic neuron. 21. The receptor, if it has a channel, is called a channel-linked receptor, or an ionotropic receptor. Ionotropic means fast response. 22. There are two types of ion channels, cation channels and anion channels. 23. Neurotransmitter-gated ionic channels flux multiple ionic species, especially the ligand-gated cation channels. 24. Calcium is a divalent cation, sodium is a monovalent cation. 25. Calcium and sodium go in the postsynaptic neuron, potassium is leaving the postsynaptic neuron, all through the ligand-gated cation channels. Multiple cations move through the same channel, even the anion channels only flux singular ionic species at a time. 26. Voltage-sensitive ion channels flux only one ionic species at a time. 27. Fast response goes through ionotropic receptors or ion channels with a passageway. 28. Slow response occurs on a timescale of seconds, instead of milliseconds for fast response. 29. G-protein linked receptors do not have a passageway and are not ion channels. The receptor is associated with another protein called a G-protein. 30. The G-protein has three subunits, the alpha, beta, and the gamma subunits. The focus in this class is on the alpha subunit of the G-protein. All three subunits are individual proteins and have their own unique functionality. 31. When the neurotransmitter binds to the G-protein linked receptor, the receptor is activated, which activates the G-protein. The alpha subunit, once it's activated, can go on to open an ion channel. It can also induce the closure of an ion channel. 32. The G-protein linked receptor is also called the G-protein coupled receptor. 33. The slow response of the G-protein linked receptor is called direct coupling, meaning the G-protein directly leads to a response by opening an ion channel. 34. The alpha subunit often interacts with another protein like a membrane-bound enzyme, or a transmembrane protein acting as an enzyme. 35. The neurotransmitters that are water-soluble need a G-protein linked receptor to utilize the alpha protein to go to a transmembrane protein acting as an enzyme to produce a secondary messenger in the cell that the neurotransmitter cannot get to itself. 36. Direct coupling is the alpha subunit activating an ion channel itself. Indirect coupling is the alpha subunit activating a transmembrane protein acting as an enzyme that produces a secondary messenger that activates an ion channel. 37. Slow response is mediated by metabotropic receptors. Metabotropic receptors are not ion channels, or they lack a channel, they're G-protein coupled receptors. 38. Once the neurotransmitter binds to the receptor, it can change the resting membrane potential of the postsynaptic neuron. Depolarizing is known as excitatory signals or EPSPs, done by channel openings. 39. Neurotransmitters acetylcholine, glutamate, ATP, and serotonin all have receptors that are ion channels or ligand-gated cation channels. Their fast response induces depolarization in the cell. This interaction generates EPSPs in the dendrite. The EPSPs move towards the axon hillock. 40. EPSPs can be made through a slow response through a G protein-linked receptor. 41. The secondary messenger from the transmembrane protein enzyme is called cyclic AMP. 42. The secondary messenger induces a potassium ion channel to close, which also depolarizes the cell. Potassium effluxes when the potassium ion channel is open. When it's closed, potassium not leaving depolarizes the cell. 43. EPSPs can be made by channels opening and channels closing. 44. Every time there is an increased positive charge on the inner leaflet of the membrane, there is a depolarization effect taking place. 45. IPSPs include ion channels that flux only chloride through passive transport. The neurotransmitter can be GABA or glycine. 46. GABA is released by neurons in the brain. Glycine is released by neurons in the spinal cord. 47. A net accumulation of negative charge is making the inner leaflet even more negative than before, from negative 70 mV to negative 90 or 100 mV, known as hyperpolarization. IPSPs make it less likely the axon hillock will fire action potentials. 48. There are two ways to make both EPSPs and IPSPs. 49. Chloride accumulation in the cell needs to be cleared. The cell spends ATP to pump chloride back out of the cell to bring back homeostasis. 50. The neurotransmitter binds to an ion channel. The ion channel that fluxes only potassium moves potassium out of the cell, hyperpolarizing the cell through potassium efflux. This is another way to generate IPSPs. 51. IPSPs can be generated by potassium efflux or chloride influx. 52. The mini brain of the neuron is the axon hillock is part of the cell body connecting the cell body to the axon. It can integrate or add all incoming EPSPs and IPSPs. And it will compare that with the threshold potential at negative 55 mV. 53. The axon hillock can add on a timescale called temporal summation, or it can add by spatial summation, or coming from different parts of the postsynaptic neuron. 54. Temporal summation is the axon hillock adding EPSPs and IPSPs from the same neuron over time. 55. Spatial summation is the axon hillock adding EPSPs and IPSPs from different neurons at the same time. 56. Temporal summation and spatial summation can be done at the same time by the axon hillock. 57. Temporal summation allows the same neuron to fire multiple EPSPs or IPSPs at the same time in order to reach the threshold potential to generate an action potential. 58. Spatial summation is multiple neurons firing. The addition of EPSPs and IPSPs at the same time can reach the threshold potential. 59. Frequency coding means how many electrical signals are being generated per unit time. 60. Presynaptic modulation occurs in the presynaptic neuron. The presynaptic neuron, once affected, will also affect the postsynaptic neuron. 61. The axon terminal of the presynaptic neuron in presynaptic modulation interacts with the presynaptic neuron next to it. In presynaptic facilitation, the presynaptic modulation neuron induces the one next to it to produce more EPSPs. The same goes for presynaptic inhibition, however it's induced to produce less EPSPs instead. 62. Aquahanic synapses are the name for the presynaptic facilitation and presynaptic inhibition. 63. Once a neurotransmitter has done its job, the messenger must be terminated in order for the neurons to recognize a signal it has to be new. 64. There are three mechanisms to get rid of old chemical signals. The first is done by enzymatic degradation, like acetylcholinesterase, which cuts acetylcholine into two pieces, acetic acid or acetate and choline. The choline is recycled by the axon terminal that releases it. 65. Presynaptic reuptake is another mechanism to get rid of old chemical signals. The neurotransmitter is recycled by the axon terminal of the presynaptic neuron. Prozac is a mechanism to prevent serotonin neurotransmitter being recycled, helps alleviate depression. 66. The last mechanism to get rid of old chemical signals is spillover. The neurotransmitter spills out of the synapse. The neuroglia will pick up the neurotransmitter to cleanse the site. Chapter 9. CNS Higher Brain Functions. 1. CNS stands for Central Nervous System. 2. Neurology is 50 years behind cardiology. 3. Reflex are the smallest structural and functional unit for the nervous system. It takes at least 2 neurons to form a reflex. 4. Involuntary motor pathways are also subconscious motor pathways. They target skeletal muscles. 5. Plasticity allows the brain to learn something, to recognize a signal and acquire it. 6. There are endocrine reflex and neural reflex. 7. The simplest and smallest neural reflex is a monosynaptic reflex. It has only 2 neurons, a sensory neuron and a motor neuron. 8. The integration within the monosynaptic reflex occurs at the synapse. 9. Integration can occur between 2 neurons. 10. Aging decreases the number of neurons. The number of neurons cannot increase, they cannot divide. 11. New synapses are created as people age. Even though the number of neurons decreases, the number of synapses increase with age proportionally to the amount of knowledge acquired. 12. Polysynaptic pathways require more than 2 neurons. 13. The sensory receptor can be a neuron or a receptor that is synapsing or making contact with a neuron. 14. Afferent neuron is the same as sensory neuron or ascending neuron, which conveys sensory information toward the integration center that's located in the spinal cord or the brain. 15. The integration center sends out a command or motor decision that is transmitted through an efferent neuron or a descending neuron or motor neuron. 16. The efferent neuron goes and makes a junction with an effector organ like a skeletal muscle. 17. The patellar reflex is hitting below the knee at a doctor's office to check the reflex. 18. The quadriceps femoris is opposed by the hamstrings group. Muscles rarely work in isolation. Every single movement has an agonist and an antagonist. 19. The patellar reflex is to test if the spinal cord at that level is still intact. 20. The patellar reflex relies on both monosynaptic pathways and polysynaptic pathways. 21. Sensory neuron is called a first-order neuron. The motor neuron is called a lower motor neuron. The synapse is specifically located in the anterior gray horn of the spinal cord. 22. The anterior gray horn is also called the ventral gray horn. However, ventral does not apply because humans walk on 2 legs, relevant to dogs that walk on 4 legs. 23. The monosynaptic pathway is utilized to contract the quadriceps femoris. 24. The association neuron is with a lower motor neuron targeting the hamstrings, part of the polysynaptic reflex. 25. The lower motor neuron that innervates the quadriceps femoris is different from the lower motor neuron that innervates the hamstrings. 26. Additional reflexes all imply using multiple neurons, still talking about polysynaptic neurons. 27. Withdrawal reflex is like stepping on a nail and the foot raises by reflex. 28. The crossed extensor reflex involves 2 limbs. One limb needs to do one thing while the other limb does the opposite or the same. 29. The voluntary motor pathway has the most important pathway called the pyramidal tract, originates in the primary motor cortex. 30. The primary motor cortex is where all the voluntary movements of the skeletal muscle originate. 31. The pyramidal tract consists of the upper motor neuron and the lower motor neuron. 32. The cell body or the soma of the upper motor neuron resides in the primary motor cortex, or the precentral gyrus. 33. The postcentral gyrus is the primary somatosensory cortex receiving information. 34. The upper motor neuron goes from the precentral gyrus to the spinal cord to synapse with the lower motor neuron. 35. The cell body of the lower motor neuron resides in the anterior gray horn of the spinal cord. 36. The lower motor neuron sends out an axon that synapses with the skeletal muscle fiber, making a neuromuscular junction. 37. The pyramidal tract allows people to voluntarily control skeletal muscle contractions. 38. The pyramidal tract controls distal extremities, the upper and lower limbs, and the fingers, all under independent voluntary control. 39. Rubrospinal tract is in addition to the pyramidal tracts. It originates in the nucleus in the midbrain, synapses in the same area as the pyramidal tracts. From the primary motor cortex it synapses. It is involved in controlling the skeletal muscle. However, the rubrospinal tract is not as important as the pyramidal tract. 40. The neurons coming from the motor cortex synapse with the neurons in the red nucleus. This is why they target skeletal muscles. 41. The rubrospinal tract is an extension of the pyramidal tract. 42. The ventromedial pathways is a collection of 3 descending pathways on each side of the spinal cord. They are called vestibulospinal, tectospinal, and reticulospinal tracts. 43. Vestibulospinal tract is originating in the vestibule, specifically the vestibular nuclei in the medulla, and it ends in the spinal cord. This tract is important to maintain posture and balance. 44. The tectospinal tract originates in the superior colliculi, is involved in visual reflexes, the eyes track any motion in peripheral vision. 45. Reticulospinal tract is from the reticular formation either in the pons or the medulla, is used for posture. 46. All 3 ventromedial pathways control posture and balance in large muscle groups near the body trunk, not in the limbs. 47. The lateral pathway controls voluntary movement. 48. The ventromedial pathway has to work with the lateral pathway in order for people to carry out daily motor function tasks. 49. Learning means people acquire new information. 50. There are 2 types of learning, associative learning and non-associative learning. 51. Associative learning means people learn how to connect or associate relationships between 2 or more different stimuli. 52. Non-associative learning involves repetition. Stimuli needs to be repeated in order to learn. The repetition will lead to 2 scenarios, habituation and sensitization. 53. Habituation is learning when there's a non-dangerous nature to something, the brain will ignore it. 54. Sensitization is learning that something is potentially dangerous. 55. Learning something needs to be stored somewhere, it's known as memory. 56. There are 2 types of memory, categorized by psychologists. There is procedural or implicit memory and there's declarative or explicit memory. 57. Procedural or implicit memory is learning how to do something like biking, becomes difficult to forget. This memory involves the cerebellum, the basal nuclei and the pons. 58. Declarative or explicit memory is forgotten over time. This memory involves the hippocampus, the memory of learned experiences like facts or events. 59. How humans acquire new information despite the incapability of creating new neurons, new synapses are created. 60. The more the information is utilized, the stronger the synapse becomes. 61. Each vesicle of the axon terminal contains hundreds to thousands of the neurotransmitter molecules in the presynaptic neuron. 62. There are thousands of dendrites on the postsynaptic neuron. 63. Glutamate is an excitatory neurotransmitter, will induce the formation of EPSPs by the postsynaptic cell. 64. AMPA receptor and NMDA receptor is a neurotransmitter-gated ion channel or ligand-gated ion channels or ligand-gated receptor or neurotransmitter-gated receptor. This is a transmembrane protein with a passageway. When they open they flux ionic species. 65. NMDA receptor and AMPA receptor has glutamate bind to both. 66. When glutamate binds to AMPA receptor, the pore opens and sodium fluxes in, causing a depolarization signal from the positive charge on the inner leaflet of the cell. 67. Glutamate also binds to the NMDA receptor, the pore opens, but the cation magnesium is blocking the channel in the NMDA receptor. The NMDA receptor fluxes a massive amount of calcium. But it cannot get in because of the magnesium cation, there's no flux because of the blockage. 68. The NMDA receptor magnesium cation can be kicked out of the channel if there's more neurotransmitter glutamate released, more receptors open, there's a stronger depolarization inducing magnesium to be kicked out of the pore, allowing calcium to be fluxed in. 69. The greater depolarization of the cell membrane induces magnesium to leave the pore within the NMDA receptor. Once magnesium is kicked out, there's calcium influx. The calcium influx is used as a second messenger. Or it can mobilize other additional second messengers. 70. A paracrine stimulus goes back to the presynaptic cell from the postsynaptic cell to induce more neurotransmitter release. 71. Long-term potentiation is the synapse being strengthened long after the initial stimulus is gone. The more the knowledge is used, the stronger the synapse becomes. 72. How the brain consolidates short-term memory into long-term memory is not understood. The hippocampus is involved in it, however. 73. Plasticity is the brain's capability to learn new concepts despite having limited neurons. It can create and strengthen new synapses instead even when the initial stimulus is no longer there, can happen for up to 2 to 3 weeks.